I'm going to continue with Oneness versus the 1% by Vandana, Shiva and Kartikey, Shiva. The case of the CRISPR. CRISPR has been described as a relatively easy way to alter any organism's DNA, just as a computer user can edit a word in a document. A 2015 article in Forbes magazine explains. 4 years ago, the protein called CRISPR-Cas9, an enzyme that bacteria use to attack viruses that infect them, was known to humans. Now it is ubiquitous in science labs as the most efficient way yet invented of a cutting and pasting DNA. Wired magazine is a breathless cover story just called it the Genesis Engine, instructing readers to buckle up because the easy DNA editing CRISPR will change the world. However, a new study published in Nature Method revealed that CRISPR introduced hundreds of unintended mutations into the genome of mice. It found more than 1,500 single nucleotide mutations and more than 100 larger deletions and insertions. None of these DNA mutations were predicted by the computer algorithms that are widely used by researchers to screen the genome of an organism to look for potential off-target effects. As of now, it is technically not possible to make a single or only a single genetic change to a genome using CRISPR, and to ensure that it has done so, as Franziska Fichtner notes, in mammalian systems, Cas9 causes a high degree of off-target effects. And as Jonathan Latham cautions, ordinary CRISPR can induce mutations at sites that differ by as many as 5 nucleotides from the intended target. In other words, CRISPR may act as an unknown site in the genome where it is not wanted. The third error that CRISPR makes is to imply that changes to gene function can be presumed to be discrete and constrained. The concept of the precise editing of a genome leading to a precise biological outcome depends heavily on the conception that genes give rise to simple outputs. This is the genetic paradigm taught in school. It is also the paradigm presented to the public, one that has been plays a large role in the thinking of molecular genetic researchers. However, a defined discrete or simple pathway from gene to trait probability does not exist. Most gene function is mediated merely through highly complex biochemical and other networks that depend on many conditional factors, such as the presence of other genes and their variants, on the environment, on the age of the organism, on chance, and so forth. Genetics of molecular biologists, however, ever since the time of Gregor Mendel, have striven to find or create artificial experimental systems in which environmental and any other system of variation are minimized so as not to distract from the more important business of genetic discovery. But by discarding organisms or traits that do not follow their exceptions, genetics and molecular biologists have constructed a circular argument in favour of a naive deterministic account of genetic function. Their paradigm habitually plays down the enormous complexities by which the information passes in both directions between organism and their genome. It has created an immense and mostly unexamined bias in the default public understanding of gene and DNAs. This indicates just how unreliable the assumptions and projections of genome editing techniques like CRISPR are, and how unreliable the claims that they are precise, predictable and therefore safe and not in need of biosafety regulation. Bill Gates and 13 others have invested $1.2 million in a CRISPR-based new company, Editas, founded by Feng Zhang of the Massachusetts Institute of Technology. MIT and the Broad Institution. The other investors are Deerfield Management, Viking Global Investors, Fidelity Management and research companies fund the account managed by T. Rowe Price Associates, Google Ventures, Jennison Associates on behalf of certain clients, Colshire Ventures, ECOR1 Capital, Casting Capital, Amiga Funds, Cowden Private Investments and Alexandria Venture Investments. The lead investor is a newly created firm called BNG Zero, a select group led by Boris Nikolic, who was previously a science advisor to Bill Gates. Both Editas and Gates' office confirmed that the Microsoft billionaire is among the BNG Zero backers. Gates also funds Jennifer Doudna, who co-founded Caribou Bioscience, to work on research users of CRISPR-Cas9, and more recently set up Intellia Therapeutics to work on disease treatments. Doudna and Editas are engaged in patent battle on CRISPR technologies, with Gates funding both sides involved in the patent battle. No matter who loses, Gates wins, and he is directly investing in corporations taking out patents on CRISPR. The patent application for CRISPR by the University of Harvard includes a long list of over 50 weeds and almost 200 herbicides that the technology could be used against, thereby laying out a business case for licensing the patent to major agrochemical companies. Editas Medicine, which has licensed one of the patents in question, disclosed that as of 2016 it has spent $10.9 million on legal fees incurred by the Broad Institute of Harvard, mostly to defend patents awarded for CRISPR invention by Feng Zhang. That cost is over and above $4.7 million spent in 2015. In a statement, the company said investing in intellectual property is one component of how we are building the company to be a leader of genome medicine, adding that it reimburses Broad and Harvard for expenses associated with the prosecution and maintenance of the patent rights that we license for them. Further, Bayer, a $96 billion German drug company, will provide the basal-based CRISPR therapeutics with a minimum of $300 million over the next 5 years. From 2016 to develop the CRISPR-Cas9 gene editing technology to treat conditions such as blood disorder, blindness and congenital heart disease. Bayer is also acquiring a majority stake in the CRISPR therapeutics for $35 billion. Bayer Corp Science joins AstraZeneca and Nestlé Health Science as strategic innovation partners in Flagship Ventures Fund V, which will focus on innovations in healthcare and sustainability in the energy, water, agriculture and nutrition sector. The ethical issues surrounding CRISPR are significant, a New York report says. Scientists are worried that it could also be used to alter genes in human embryos, sperm or eggs in ways that can be passed from generation to generation. The prospect raises fears of a dystopian future in which scientists create an elite population of designer babies with enhanced intelligence, beauty and other traits. Sequences are frequently repeated within a genome, often as an artifact of evolution, and scientists have expressed the concerns that there is mounting evidence that gene editing can have unpredictable off-target effects, i.e. it may cut and replace not only the target gene but other genes with the same partial sequence somewhere in the genome. Finally, Ricardo Steinbrecher notes that in some countries CRISPR is falling into regulatory void. the US Department of Agriculture, USDA, declined to regulate a CRISPR-engineered mushroom in 2016, saying it was outside USDA's biotech regulations. CRISPR has become the window for the biotech industry to continue the failed GMO experiment. Monsanto has signed an agreement for agriculture applications of CRISPR with the Broad Institute. Bill Gates, biodiversity, gene drives and new GMOs for the Terminator to the Exterminator. DARPA, the Pentagon Research Arm, and Bill and Melinda Gates Foundation sponsored in 2016 report by the National Academy of Sciences, NAS, in the United States, titled Gene Drives on the Horizon, Advancing Science, Navigating Uncertainty and Aligning Research with Public Values, which warns one possible goal to release of a gene drive modified organism is to cause the extinction of the target species or the drastic reduction in its abundance. Gene drives have been called mutagenic chain reactions and are to the biological world what chain reactions are to the nuclear world. The Guardian describes gene drives as the gene bomb. Kevin Esvelt of the MIT exclaims, "A release anywhere is likely to be a release anywhere," and asks, "Do you really have the right to run an experiment where if you screw up, it affects the whole world?" The NAS report cites the case of wiping out amaranth, an example of a potential benefit, yet the magical technology of gene drives remains a ghost, or the US government's Department of Defense secret weapons to continue its war on Amarantha's culatrice. Some 75 years ago, DARPA Mind began its extermination experiment and sent humanity off axis. DARPA Mind is our label for the militarized mind. The chemical materials and technologies acquired during the war and patented were forced on Amarantha's culatrice. The following objective and rationale have been stated in the NAS report. Objective: create gene drives in Parma amaranth, Amarantha's palmaria, also called pigweed, to reduce or eliminate the weed on agricultural fields in the southern United States. Rationale: Parma amaranth infests agricultural fields throughout the American South. It has evolved resistant to the herbicides glyphosate, the world's most used herbicide, and this resistance has become geographically widespread. Parma amaranth has emerged as one of the world's superweed instead of seeing its emergence as a superweed as a result of the failure of misguided herbicide resistance. GMOs, Monsanto and Co. investors, scientists, corporations, DARPA and Gates are now rushing to drive the amaranth species to extinction through the deployment of an untested tool. The NAS report casually states the potential human harm to gene drives. Gene drives developed for agricultural purpose could also have adverse effects on human well-being. Transfer of a suppression drive to a non-target wild species could have both adverse environmental outcome and harmful effects on vegetable crops. For example, Parma amaranth in case study 6 is a damaging weed in the United States, but related amaranth species are cultivated for food in Mexico, South America, India and China. For the high slopes of the Himalayas through the plains of North, Central and South India to the coastlines of the East, West and South, amaranth is a web of life. In fact, the Himalayan region is one of the centres of diversity for amaranth. Its leaves contain more iron than spinach and have a much more delicate flavour. Apart from rice bran and grain of amaranth has the highest content of iron among cereals. 1 gram of amaranth flour added to 1 kilogram of refined wheat flour increases its iron content from 25 milligram to 245 milligrams. Adding amaranth flour to wheat rice flour is a cheaper and healthier way to prevent nutritional anemia rather than buying expensive tablets, tonics, health drinks, branded and biofortified flour or canned spinach. The amaranth is extremely rich in complex carbohydrates and proteins too. It has 12 to 18% more protein than other cereals, particularly inosine as critical amino acid. It also differs from other cereals in the 65% of it is found in the germ and 35% in the endosperm compared to an average of 15% in the germ and 85% in the endosperm in other cereals. The amaranth grain is also the richest source of calcium other than milk. It has 390 grams of calcium compared to 10 grams in rice and 23 grams in refined flour. Industrial agriculture treated amaranth greens as weed and tried to exterminate it with herbicides. Then came Monsanto's Roundup Ready crop, genetically engineered to resist the spraying of Roundup so that the GMO crop would survive an otherwise lethal chemical, while everything else that was green perished. Financialization, patentisation and forced digitization. In the 21st century, the corporate empire has found new instruments of extraction with patents and what should be called capitalization being the two most prominent ones, while the chemical biotech seed industry innovated and invented patents on life and on seeds as a means to extract profit from farmers. The second instrument, financialization of the economy, is equally perverse. It has marginalized the real wealth that people produce in the real economies and primarily rewarded the capitalists. The financial economy is now 70 times bigger than the real economy in the world. The concentration of the world's wealth in the hands of the 1% is a consequence of patentization, financialization and digitization of our lives. The most recent bait and switch has been used by globalized corporate power to worm its way into households in India is the hidden demonization of large denomination notes. Towards the end of 2016 was a concerted and conceived attempt to impose a digital economy on the country through a cash ban. Those without smartphones and credit cards were overnight converted into digital barbarians, needing to be civilized and mainstreamed through digital literacy. The demonization of India's economy at the stroke of midnight on the 8th of November 2016 was a gamble by corporates to appropriate people's wealth by locking it behind an encryption key, shutting down people's economies overnight by allowing only those sections of the economy willing to indulge in payment gateway keepers to operate. NASPER's group is that gatekeeper for digital India. More than 90% of India functions on cash to sustain the people's economy. By withdrawing 86% of all currency overnight and declaring it illegal, the damage inflicted on the majority of ordinary Indians was colossal, just as patents on seeds were an illegitimate attempt to criminalising farmers by making seed saving illegal. Demonization was an illegitimate attempt at criminalising people's savings and seriously disturbing their economy, which compromised 80% of Indians' real economy. Bill Gates, in his speech for the NITI lecture series Transforming India delivered in the presence of Prime Minister Narendra Modi and his cabinet colleagues, stated the government's bold move to demonetise high-value denominations and replace them with new notes and high security features was an important step to move away from the shadow economy to a more transparent economy. He's further said that digital transactions would rise dramatically as a result, and in the next several years make India one of the most digitalised economies, not just in size but by percentage as well. So I'm going to leave it there for now.